Hello, today we will discuss on Voges Proskauer test, which is commonly known as VP test. So before we go to the procedure, let me tell you briefly about this VP test. So this VP test is mainly to differentiate two major types of facultative and anaerobic enteric bacteria based on production of neutral product. So this Voges and Proskauer in 1898 they first observed the production of a red color after the addition of potassium hydroxide to culture grown on specific media. Later on, Harden reveals that the development of the red color was because of the acetyl methylcarbinol production. Later in 1936, Barrett made the test more sensitive by adding alpha naphtol to the medium before adding potassium hydroxide. Therefore, in this VP test, we are going to be using the two reagents, that is potassium hydroxide and Barrett's reagent. This we will discuss in the later part, in the procedure or in the reagent part. So let us see the principle of this Voges Proskauer test. So this Voges Proskauer test, commonly known as VP, is used to determine the if an organism produces acetylmethylcarbinol from glucose fermentation, and in if present acetylmethylcarbinol is converted to diacetyl in the presence of alpha naphtol, strong alkali, and atmospheric oxygen. But here let me tell you briefly about this principle of this VP test. So basically in a very simple way I'll explain it to you. So when we inoculate this bacteria into glucose phosphate, here because we are going to use the glucose phosphate broth again, which is used for the MR test as well. So when this bacteria is inoculated into this broth, glucose phosphate broth, the bacteria converts glucose phosphate into a pyruvate acid. Now, we all know that this pyruvate acid has different metabolic pathway, so bacteria by some other pathway will convert this pyruvate acid into acetone, which like after an incubation period at 37 degrees centigrade for 48 degree for 24 to 48 hours. Now, here, after the incubation period, we have to detect the acetone by adding the 5% alpha naphtol reagent and then 40% KOH. And the addition of this reagent has to be done in the presence of the oxygen only. And also remember this reagent should be added accordingly only. So this is the principle of the Voges Proskauer test. Now let us see what are the reagent and the materials we require to perform the VP test. Here we are going to use the MRVP broth again, which is used for the MR test, methyl red test. Then here Voges Proskauer reagent A, the also called as Barrett's reagent A, we are going to use that, and Voges Proskauer reagent B, Barrett's reagent B. So here I have given the constituent of both the Barrett's reagent A and Barrett's reagent B. So here in the given picture, the above the down one is the Barrett's reagent, whereas the above one, the up one, is the MRVP broth, which is a glucose phosphate broth. Now these are the media and the reagents we are going to be needing for performing the VP test as well as for the MR test. Let us see the procedure of this VP test. So the procedure is prior to the inoculation of the bacteria into this glucose phosphate broth, we have to allow the medium, we have to allow this broth to equilibrate to the room temperature. So most probably before we start the practical, we have to take out the broth and keep it in the room temperature so that it gets equilibrate with the room temperature. Now after that, we have to, with the help of the sterile inoculating loop, we have to take the organism from an 18 to 24 hours pure cultured, and then inoculate in the medium, after which we are going to incubate aerobically at 37 degrees centigrade for 24 to 48 hours in ambient air. Now after the incubation of 24 to 48 hours, you are going to add 6 drops of 5% alpha naphtol, and we have to mix well to aerate. Again, we have to add 2 drops of 40% potassium hydroxide and mix well to aerate. Remember, this alpha naphtol, 5% alpha naphtol and 40% potassium hydroxide KOH, this have to be added in the presence of oxygen only and in accordingly. Because if suppose like you have, instead of adding 5% alpha naphtol in the first place, if you add 40% potassium hydroxide then add 5% alpha naphtol, it might give some false positive interpretation. Now after the addition of these two reagents, you are going to observe for a pink red color at the surface within 30 minutes. So it should be interpreted within the 30 minutes, not more than that. Then you have to shake the tube vigorously during the 30 minutes period. So when you shake it, when you shake the tube, make sure that you gently shake the tube. Now coming to the interpretation here, the positive reaction, it is indicated by a red pink color at the surface, which means the bacteria has converted this acetone into red color compound. That is why it has that is why your test organism can be reported as a VP test positive, Voges Proskauer positive. Whereas here in the negative reaction, there is the medium remain unchanged, the color remain unchanged. See here in VP negative, the broth is still yellow in color, whereas in the positive it has already turned into a pink color. This is because the bacteria has converted into an acetone. So basically, when your medium does not change the color, this means the bacteria has not converted the acetone, and therefore your broth remains colorless or remain yellow in color. So in this case, you have to interpret your result as the given organism is not an indicate is a VP negative test. Now coming to the quality control, again here the bacteria we have, why do we use quality control, is that we should regularly test to check the reagent, and also to test the reagent and also to check the potency. So some of the suggested controls are: for VP positive we have Enterobacter aerogenes, then ATCC 13048. Then for VP negative we have Escherichia coli ATCC 25922. So these are some of the suggestive quality control for the VP test. Some of the examples of VP positive are Klebsiella, then Viridien group Streptococci, then the Vibrio species, and the negative VP we have Shigella and Citrobacter. Now again, we have some limitation for VP test. So this limitations are like: this VP test may be used in the identification of gram-negative bacteria, on the addition of biochemical testing using the pure culture, which is recommended for the complete identification. Now, very important, the order of the addition of the reagent is very extremely important, as I have mentioned earlier also. In case in the reversal of the order may result in a weak positive reaction or false negative reactions or interpretation. And also this MRVP testing should be used in conjunction with other confirmatory tests to differentiate organism among the Enterobacteriaceae. So this is all about the VP test.